Okay, boys and girls, it is time to get our lecture on. All right, we're going to be winding up this semester with Chapter 10, the humoral immune response, okay, because this course is humoral immunity. We're going to be looking at what it takes, what this humoral immunity is all about. So, is some microbe, something not you, has bound to B cell receptors. It's activating the B cell, but it's going to need a T helper cell. Remember the multiple types of T cells? T helper cells are going to come along, and there's going to be ligand receptor interactions. CD40 ligand on the surface of the T helper cell, CD40 on the surface of the B cell. They're going to go back and forth. Cytokines are going to activate the B cell. It's going to tell it to start secreting as much antibody as possible. Well, to start this off, first thing that's going to have to happen is something not you. An antigen is going to bind the B cell. It's going to bind, and it's going to start also recruit in the T helper cell. Now, when you look up here closer, so first up, antigen, whether it's on the surface of a microbe or some sort of parasite, it's going to bind here, and it's going to begin activating the cell, but it's not a one step and done. It's also going to need this secondary signaling through the CD40 ligand. This ligand activation will help bring about cytokine production. Cytokines binding their receptor will then activate the B cell to start expression of antibody genes. Now, this top one here, looking at the T helper cell, this is what is known as thymus dependent. Thymus dependent means it has to have a T cell. Okay, remember the T cells mature for a while in the thymus, hence T cell, T thymus. There's also an independent. Sometimes antigens are large enough. They can bind to multiple B cell receptors, co-opt quite a few of them, or you get something like LPS that binds a toll-like receptor. Remember, LPS is a super antigen, and if it binds, it just activates. So here, something big has bound up quite a few of the B cell receptors. Signal 1. Signal 2 is LPS binding the toll-like receptor. So we get to bypass this whole CD40 ligand and cytokines, and we just go straight through without T cells. Now, turns out the thymus dependent activation actually it has a higher rate of activation potential, has a higher rate of getting B cells active, getting them to switch from being quiescent to being a plasma cell. It turns out that not only the CD40 ligand, but it's the IL-456. This is a push that will push the B cell to go through rounds and rounds of proliferation. This push, basically it's like a weight falling on the B cell. It's just going to start knocking over the dominoes. We're going to start the signaling cascades that's going to tell the cell to proliferate. One's going to become dozens, hundreds, through rounds and rounds of mitosis. Now, not every one of these cells is going to become a plasma cell. Some of them are going to stay a memory cell, remain quiescent, and wait. Wait for more T cells, wait for another cytokine push to get them to go. Now, this is not a willy-nilly process, okay? Turns out that the B cell is getting ready to be activated by a T cell. Well, that T cell has to also be able to recognize the same antigen. So there is a linked recognition. If you look here, the MHC class 2 that has an antigen loaded in it is also interacting with the T cell receptor. So for that interaction to occur, for that interaction to be stable enough for the CD40 ligand as well as the cytokine production to occur, that antigen that's holding all that together at the center there has to be recognized by MHC as well as the T cell receptor. So they call this a cognate T cell. It's not just a random T cell coming by, it is a T cell that is also recognizing the same antigen. This is a step to ensure that autoimmunity doesn't take place. Okay, this helps us maintain self-tolerance, because if this was a random T cell could do this, well, what if that random T cell just happens to activate a B cell that has a self-antigen? It's not making self-antibodies, but it has a self-antigen. So it's going to, start responding. And maybe it is, a B cell that's making not quite self, but almost self-recognizing antibodies. Well, this will push it closer and closer to being an autoimmune issue. So, some sort of, here they're showing a virus coat protein. So a virus binds to the B cell receptor, gets phagocytized, digested down, and an epitope, an antigen, gets loaded into MHC. MHC class 2 is presented on the surface. A helper T cell recognizes MHC. It's an unstable interaction until everybody changes conformation, and then the antigen, the epitope, is now stabilizing the T cell receptor and its co-receptors there, which brings in CD40 ligand to interact with CD40, and that signals the T helper cell to make a bunch of cytokines. Cytokines which are then going to be the final push for the B cell to become a plasma cell and start making antibodies. Now, that was an example looking at virus proteins, but it turns out that bacterial polysaccharides, these are going to be the surface sugar moieties, sugars that may have proteins or amino acids on them. These are going to be the surface molecules on bacterial cells. They can also go through the same process and recruit and activate helper T cells. It's this interaction that we utilize for many of our vaccines. Okay, we're not going to inject somebody with, strep pneumonia or things like that. We're going to inject them with a digested, broken down bacterial cell. Trace quantities of it so that B cells can bind the B cell receptor, take them up, digest down, and then T cells can be taught. T cells can mature towards that antigen, that epitope also, and now, you're good to go. So if you look at this, it looks quite similar to what we were just talking about with the virus. Binds the receptor, taken up, digested, loaded into MHC class II, because remember, it is a vesicle. It's in the phagolysosome. It's not in the cytoplasm. Expressed on the surface, recruits in helper T cell, interaction T cell receptor, MHC class II. It's not stable until you also now get the epitope, which stabilizes T cell receptor, co-receptor, brings in CD40 ligand interaction, and then cytokine production, and you get opsonizing, neutralizing antibodies produced.